Hello, everyone, and welcome to phyloseminar.org. Sorry about the technical difficulties there. So phyloseminar.org is a project supported by the Society of Systematic Biologists. In this series of 3 talks, we're looking at phylogenetic opportunities and challenges from bacteria and archaea. Our first talk was from Laura Hug about recent discoveries of new groups of organisms. Then we heard from Xavier Didelot, and today we are hearing from Tim Vaughan. I want to remind people that the Q&A app is gone now, so you can ask questions through Twitter or IRC, as described on the attending section of the Phyloseminar website. As I mentioned, today's speaker is Tim Vaughan. Tim got his PhD in theoretical physics in 2008 from the University of Queensland. Directly after his PhD, he spent a year in Jeff Goodhill's neuroscience lab as a research assistant. He then started working with Peter Drummond and Alexei Drummond. So Peter is Alexei's dad, modeling within-host viral dynamics, which led to a bunch of visits to Auckland, where he discovered phylogenetics. He's now a postdoc in the computational evolution group at the University of Auckland. Welcome, Tim, and thanks for participating.
Thank you very much for that lovely introduction, Eric, and thank you for giving me the chance to speak to everyone today. I'm just going to start my slide sharing. Okay, so yeah, thank you very much for that, Eric. My name is Tim Vaughan, as Eric just pointed out. I'm going to be talking today about some work I've been doing on joint Bayesian inference of bacterial ancestral recombination graphs. And this follows directly on from Xavier Didelot's lovely talk a couple of weeks ago. Because a lot of my work has been involved with implementing those models in BEAST. I'll talk a bit more about that. So firstly, why would we want to study bacterial phylogenetics? Well, there are a number of really good reasons. Bacteria obviously play very important roles, both positive and negative, in the health of many animals and plants. From a scientific point of view, they also possess many very interesting and experimentally accessible evolutionary dynamics. This is really important, especially for phylogenetics, because this allows us potentially to test rather directly the inferences that we draw from genetic information. Furthermore, bacterial, and this goes along with being experimentally accessible, bacterial genomes are measurably evolving, quote unquote, in the sense that you can expect to see real evolutionary change over a relatively short study period. So over the period that you collect your samples, you can expect to see genomic variation, especially on the genome level. So once you're looking at whole genomes, you can definitely start to see evolution as it happens. And this means that you start to enter this crossover regime between population dynamics and the evolutionary dynamics. So you can start to infer interesting things about population dynamics from the genetic information. Okay, so as Eric pointed out, my background is in physics, so I have a rather, I guess, a cartoony view of bacteria in general, probably far too cartoony, but this is basically the way I see things. You have bacteria looking like this to some rough approximation, and this bacterium reproduces clonally. So they usually reproduce by binary fission like this, and so you naturally get this tree structure emerging, and that sounds like a phylogeneticist's dream because we like to infer trees. But of course, life is never that simple, and there are a whole load of different mechanisms that make this picture a little bit messier. So horizontal gene transfer, etc., occur. Non-vertical inheritance occurs via a number of mechanisms, including phage-mediated transduction. You also get natural transformation, so bacteria are just absorbing naked DNA from their environment and incorporating it into their genomes. And they also have this weird form of sex where this conjugation process where they connect to each other with these pipes and pump genetic material into one another. I think this is crazy business going on up here. Yeah, I'm not going to talk too much about that. Okay, so the effect of these events can be plasmid transfer, insertion, or deletions, or homologous recombination. The rate of recombination varies between different species, and itself is subject to selection, which creates an interesting feedback loop. And we tend to focus on homologous recombination because basically, mostly for practical reasons, because it's the only event that really doesn't alter the length of the sequence. So you can still have a nice sequence alignment and have homologous recombination still affecting your alignment. Okay, so the problem for phylogenetic inference is that for many bacteria, the ratio between the recombination rate and the mutation rate is extremely high. Now, Vaughan and Didelot presented a very detailed study of this a few years ago in 2009, looking at the ratio of not exactly the recombination rate to mutation rate, but the R to M ratio, which is the ratio between the probability of a site being modified by a recombination versus a mutation. So it's similar to the recombination rate to mutation rate ratio. But you can see that from this, I've just thrown their results into a histogram, and you can see that the mode of this distribution is somewhere around 1. So recombination and mutation are both playing sort of very similar roles in that they're producing roughly the same amount of variation. And in some cases, recombination can produce a lot more variation than mutation. So it means it's an extremely important process for a lot of bacteria. There are exceptions, obviously, but generally this is the case. So what can you do? So there are a number of solutions that exist. Obviously, you can pre-process data sets to remove anything that you think is any variation due to non-vertical inheritance. So this is just one method of doing that. This method called start. Pros of this approach is that obviously you can then just use, in principle, standard tools for phylogenetic inference. So Mr. Bayes or Beast, and just ignore the fact that recombination is going on. Con, obviously, is that data is being thrown away. It's a lot of data is very hard won, obviously, and it's very sad to throw it away. Not only that, it's sort of an ad hoc approach, because exactly how you go about deciding what variation is due to recombination and what is not is something that's very hard to do in isolation without considering a tree at all. So trying to factor the inference into this step that involves cleaning the alignment and then doing a tree inference is always going to be worse than trying to properly model the effect of those non-vertical inheritance processes on the ancestry. So the other obvious approach is to explicitly model bacterial recombination, and this is the approach that Xavier has taken a number of times in his wonderful models and packages. The pros of this are obviously that you can start to make use of all of the data you have, and you can also, because you're actually modeling the recombination process, you can infer things like recombination rates and actually start to try to understand that process rather than try to ignore it. And interestingly, it may also be possible to, this may allow you to increase your inference power for particular parameters, specifically the population size. I'll get onto that a little bit later. The drawback, obviously, is that the models are a lot more complex than just tree models, and they have many parameters. And because of this, they tend to be inferences based on these models tend to be computationally and statistically challenging. The problem that we saw was that the existing implementations, because they were standalone implementations, lacked a degree of flexibility. And this is just a side effect of them being standalone implementations. We've tried to implement some of these things inside Beast, which means that you automatically get access to the variety of substitution models and priors, etc., that Beast already has for free. Okay, so as Xavier sort of outlined very nicely a couple of weeks ago, the process that best describes recombination in bacteria when you're interested in homologous gene conversion is the coalescent with gene conversion. So it's based on a Wright-Fisher model, which I've tried to depict here. So time goes vertically down the page, and these circles represent individuals within a given generation. And you can see that in each generation, each circle has a parent in the previous generation, and that parent is indicated by the line joining the child to the parent. So this naturally gives rise to a tree-like structure. And you can always draw this tree for bacteria, because if you're assuming that binary fission is always going on, this is actually the tree that represents which bacteria in the previous generation were parents of bacteria in the current generation. At this level, it's just the same as the regular Wright-Fisher model. So we can start to consider what happens when we sample a number of individuals from the final generation and trace their ancestry back, and that's what these blue lines represent. You see that occasionally these blue lines coalesce at these different points, and you wind up with this sampled tree. Now, with the presence of recombination, there are additional events that you have to consider. So it's possible that between these two generations, this particular individual didn't only receive genetic information from its parent, but a portion of its sequence was underwent a homologous gene conversion event. So that's what this little red bar here indicates. So this is saying that a portion of the sequence in a small range here actually came from this parent rather than this one over here. So to trace the ancestry within over these sites, you actually have to follow the ancestry of this individual, which we get just by tracing that previously black line back again, and eventually it joins up with the blue line here. And you can actually get more complicated structures, of course, as well. So it's possible that this individual itself inherited information not only from its parent, but also from this other individual in the previous generation. And so you can wind up with sort of these dashed edges on top of dashed edges representing multiple recombinations in sequence. Okay, and when you discard everything that wasn't sampled, this is what you get. And this is the ancestral recombination graph. And in the case of the coalescent with gene conversion, there's this asymmetry between the clonal frame, which is what I've colored blue here with the solid lines, and these recombinant edges. The clonal frame is what you get when you observe the entire process under a microscope and don't pay attention to the genetics really. but if you don't have a lot of recombination, this is what dominates, obviously, the genetic variation as well. But these recombinant edges are only carry a small portion of the genetic information, and they are resulting entirely from these additional events. Okay, so this model was developed well 17 years ago now by Carsten Wurf and Jürgen Hein in a couple of papers, and it's called the Coalescent with gene conversion model. So you arrive at this model by taking the usual coalescent limit, that's having very large population size compared to the sample size, and a very large number of generations. So it's a continuous limit of that process that I just described. And the parameters involved are the coalescence rate. So this is just the regular coalescent parameter. So that's actually the inverse, the coalescent rate. But so this n of t represents the population size at time t. So this can be a dynamically varying population size. G is just a generation time. Rho is the rate of recombination. So this is the per generation hazard of recombination events occurring on your tree. Recurring along lineages in the ARG. And delta is the expected track length. So that gives you the characteristic length of these fragments that come from other sources. And in the model, this is geometrically distributed with a mean of delta. So there's quite a lot of variation allowed still for these track lengths. So that's quite a broad distribution. Okay, so the problem is that the space of possible ancestral recombination graphs is extremely large. Unlike tree, and tree space is already incredibly huge, but ARG space is even infinitely larger. To give you some idea, even two samples, which would just form a cherry tree in regular phylogenetic analysis, can have infinitely many distinctly, topologically distinct ancestries associated with them. So it's quite a vast increase in complexity. And for this reason, it is really good to try to simplify the model if at all possible. And there are a couple of ways that this has been done. Both of these are due to Xavier Didelot and colleagues. So this is the full ARG here on this page. And we notice that we've got, so the clonal frame is in black, and the recombinant edges are in the dashed blue edges. And you can see that you have a number of different types of events. You've got recombinant edges that coalesce together to form a single ancestral edge. You also have recombinant edges that bifurcate backward in time. So recombinations layered on top of one another. Now, the first approximation that was made was to basically for those recombinant edges and ask where they came from, but instead just treat them as these sort of strange substitution events on the tree that affect a number of sites simultaneously. So that's the basis of the ClonalFrame model, which has been implemented both in a Bayesian format and a maximum likelihood framework more recently. The second approximation, which is the ClonalOrigin approximation, is to restrict the kinds of events that can occur. So involving recombinant edges, to say that no recombinant edge can itself experience another recombination on top of it. So there's no bifurcating recombinant edges here. And also you don't get coalescence between recombinant edges. So in other words, every recombinant edge must depart from and attach to the clonal frame itself. You can see that this means that if you're going to try to describe this full ARG, you're going to need a couple more recombination events, because this event and this event are not allowed. So you have to replace those events with some equivalent approximation. But so in this case, every recombination lineage departs from and attaches to the clonal frame. This is the basis of the ClonalOrigin software as well, and the ClonalOrigin Prime model, which is the ABC variant of this. Okay, so our goal is to come up with a way of conducting Bayesian inference on the full ARG under the ClonalOrigin model. So using this model, but unlike the ClonalOrigin software, we are interested in trying to infer this entire object jointly. The ClonalOrigin software requires that you condition on the clonal frame and infer the recombinant edges on their own, whereas we want to do it together. Now, inference under the ClonalOrigin model is quite, I mean, from a mathematical standpoint, at least, quite straightforward, and it shares a lot in common with regular phylogenetic analyses. So if you've seen a standard phylogenetic inference, Bayesian inference posterior distribution, the distribution here looks very, very similar. So you have this likelihood term where A is the sequence alignment. So this is the probability of the sequence alignment given the genealogy. So this is the full ARG, and the substitution rate. So this is different from the regular phylogenetic tree likelihood, but only trivially so, because because the effect of the recombination events is to cause you to have multiple trees that affect contiguous sites in the alignment. So if you split your alignment up into the site ranges that correspond to each of these individual trees, you can compute the regular phylogenetic likelihood of each of those trees, and just multiply them all together, and that gives you this object. This density over here is the what I would call the tree prior, or the ARG prior. It's the probability distribution of over the ARG space in absence of the sequence data. So depending only on the population size parameter and the recombination rate and tract length parameter. This object, this distribution encodes the majority of the information about the ClonalOrigin model itself. So this is the main part of ClonalOrigin, really. And then you just have parameter priors. So because because of the approximations that the ClonalOrigin model makes regarding the independence of recombinant events, you can factorize this distribution over the genealogies into an expression involving the probability of a single recombination events given the clonal tree. And this can significantly speed up computation if you have a fixed tree, but it does make things easier for when you're doing the joint inference as well. This is all just background, but the real problem is that even though we're using a simplified model, you still have an infinite number of ARGs that possess exactly the same likelihood given the sequence alignment. So this is a real identifiability issue. And to demonstrate what I mean, just consider these 3 ARGs. So I've got just 3 samples here. I've got a single recombination event that affects some portion of the sites colored red here. Now I could describe in terms of the likelihood, in terms of the probability of the sequences, in other words, in terms of the probability of the sequences given the ARG, this ARG is completely equivalent to this one here, where I have, instead of, so firstly note that the topology of the clonal frame is shifted. So actually, the topology of the clonal frame is exactly the same as the marginal tree associated with these sites here. So in other words, if you are considering sites in this region of the alignment, then to get the local tree topology, you follow this dashed edge up here, and you have a coalescence event here. So that's exactly what you have in black here. But on this side, this clonal frame applies to exactly the same region that the recombination applies to in this ARG. So you can see that there are 2 recombinations, but they affect everything outside of that small range. And their effect is to cause the marginal trees outside of that range to look exactly like the clonal frame over here. So that's a particularly nasty feature of the model. I mean, it's sort of a fact of life that you get these different ancestries that just don't produce an actual signal. But it's not really the end of the road for Bayesian inference, because we can use our priors to select between these 2 options, but it still means that in practice it becomes difficult to sample the state space. And thirdly, you also get a third equivalent ARG is where you have exactly the same ARG as the first one over here, but you've added these little loops. And these little loops are real events that occurred. There was a recombination over here, but the lineage that the donor lineage coalesces before the parent of this node here, so on the same edge in the sample tree. And as a result, if you're assuming a strict clock, which unfortunately we are, there is no signal for this sort of thing. It makes absolutely no difference to the likelihood. And we can add arbitrarily many of these, and it will not change a thing. Loops can have an effect, so it's important to include them, and also to exclude them would bias your estimates of recombination rates. But yes, in general, they often don't have any effect on the likelihood. So anyway, it's very important then for an MCMC algorithm that tries to sample this state space to propose changes that minimize the effect on the likelihood. Because this is these directions that you move in when you shift from this sort of ARG to this sort of ARG are going to be directions that the posterior is going to be quite broad in. So you really need to explore along these directions very efficiently if you're going to sample the state space in finite time. The way that we propose to do this is to use this particular strategy, and there are a number of different state proposal mechanisms built into the algorithm, but this is the one general theme that has helped a lot, I think, in exploring this joint state space. So we have an ARG here, and we want to propose a new ARG. The way that we can do it is we can detach this black edge. So this is just chosen randomly, but this solid gray edge rather is one that I'm going to delete. And I'm going to reattach it over here. So this is like an SPR or a Wilson-Balding sort of move in tree space, and it's operating on the clonal frame. But the difficulty is that we've got all of these additional recombination events that we have to somehow modify in a sensible way. And the way that we do that is to what I call collapse them. So that means that any attachment point that's on an edge that's going to be removed is moved over to a new attachment point, which is still ancestral to still ancestral to C. Let's put it that way. So you can see that this this blue dashed edge here becomes this blue dashed edge here. Along with that, you also might note that this this purple edge up here has to be removed entirely, because one of the features of this model is that you don't explicitly include recombinations that begin above the root. If you were to do that, you would always wind up with a process that did not finish as you went backward in time. So we need to delete this one entirely. And of course, that means that in the reverse direction, because all of these proposals must be reversible, you need to be able to create these edges on the fly, and also to do the reverse of this collapse procedure. That is to take an edge like this and reattach it to a new gray edge over here. It's a little bit tricky to describe exactly how this works, but hopefully that this gives you some indication of what we're trying to do. Okay, so, and all of these, anytime we have to create a new recombination edge in one of these moves, we obviously try to draw this directly from the prior. That's sort of one of the sensible things, the least, I wouldn't say it's the most sensible thing to do, but it's not completely crazy. Okay, so we have this algorithm. We've implemented it. We need to validate it. The first thing that we do to validate it is to try to sample directly from the approximate ARG via Coalescent simulation. So that's very easy to do, because Coalescent models are very easy to simulate under. And that means that we can then do MCMC. So use our sampling algorithm to explore the same prior distribution and compare the results between the simulated ARG distributions and those generated by MCMC. And we expect very precise agreement, and happily that's exactly what we get. These are just a number of different summary statistics, so clonal frame height, mean recombination edge length, and mean track length, and you can see that the MCMC matches up with simulated distributions very precisely. This is very important to do this whenever if you're implementing numerical methods. It's extremely important to have quantitative tests. Qualitative tests aren't good enough. At least they're not good enough for me, because I make mistakes when I code, and I need to have some way to detect those flaws and fix them. So I can't stress this enough if you're implementing MCMC algorithms to find a test like this. The other thing that you can do, obviously, is you can simulate some sequence data. So simulate an ARG using known parameters, simulate sequences down there, down those ARGs, and then try to infer the parameters from the simulated data. And this is another really good way of testing the implementation. But unless you do this carefully, it's not going to be a real quantitative test. You can do this in a way that means that the test is quantitative using what's known as the well-calibrated approach. We go into that in the paper, but that's not what this is. This is more of a qualitative. Yes, we can infer the tract lengths and recombination rates and population sizes. So it gives you some indication that things are working, but it shouldn't be the end of the story. Okay, parameters are wonderful, but what we're really keen to see are the ARGs. This is a representation of the true ARG that was used to generate the sequence data that we analyzed in that simulated data analysis. Now, this requires a little bit of explanation. The black lines represent the clonal frame. The dashed lines represent the recombinant edges. The numbers here represent the site ranges affected. So there's only a single locus. Oh, there are two, rather, sorry. There are two loci here, L1 and L2. And the numbers represent the sites affected on each of those loci. So that's the truth. We've got a randomly selected ARG from the MCMC output down here. And you can see there are a lot of really nice correspondences. So it has managed to find a lot of the recombinations present in the data, which is wonderful. But it's not really very convincing. This is just a single ARG selected from the posterior. How do you know whether any of these conversions have any real support? The way we've sought to answer this is to take a similar approach to the one that people often take in phylogenetics, which is to create some sort of consensus or summary tree. But in our case, we have we need to create a summary network. Now, we couldn't find any existing methods for doing this that would work with our particular networks. So we've had to sort of construct one ourselves. And what we do is we take our set of samples from the posterior. So this is just 1 and 2 different samples from the posterior. You can see that they have two recombination events that maybe are should be compared and treated as a single event, but who knows? So what do we do? Basically, we firstly just consider the clonal frames and construct a maximum clade credibility tree, so just a standard summary of those clonal frames. And then once we have the summary, we consider the recombinations that join particular uniquely identified edges in that summary tree. So in other words, we look back through all of our posterior samples and look for, in this case, recombinations that join the edge above C to the edge above the MRCA of A and B. And so we find this collection of recombinations. We look at the sites affected for each of these recombinations, and then constructs construct this graph of the number of conversions affecting each of these sites along the alignment, and then rescale this so that this is not just a number; it's the fraction of the ARGs, of the sampled ARGs that have conversions between these edges that affect the particular sites. And then we set a threshold and say that whenever we go above this threshold, we're going to produce a summarized conversion event. So this collapses down and we wind up with this single conversion event between these two edges, with the connection points determined by the means of all of the sampled conversions that are involved, and a similar process is used to select the start and endpoints. Right, and so when you do this, you construct these summary networks. You can see they're a lot less busy than the tree ARGs, and that is mostly because the conversions that have support, there are going to be conversions on the tree ARG that don't have support for precisely the reasons that I've discussed, like the conversions may not actually affect the likelihood at all, so it's impossible for the sampler to identify them precisely. That's not to say it won't be proposing the correct number of conversions. It will be, but they just won't have any solid posterior support. So the ones, the conversions that appear down in the summary ARG are the ones that that both are detected and detected with sufficient in a sufficient fraction of the posterior ARGs. Okay, so we have applied this method to some E. coli data. So we've got 23 isolates that have been sequenced and full genome sequenced, and we extracted the RMLST genes from these genomic sequences. So there's 53 of these RMLST genes, and we analyzed using a fixed expected track length parameter and a relatively informative prior on the conversion rate. It always helps if you can include any sort of prior information in these analyses. It can cut down on the computational burden associated with doing them. Okay, this is a splits tree figure of the concatenated data set, just to give you some indication that there is recombination going on, and we'd like to try to detect it using the ClonalOrigin model that we've and our implementation of it. So this is what we get. This is a summary network. The edges, recombinant edges here, are labeled with the locus that they correspond to. And you can see that there is quite a lot of recombination going on. And what's nice is that you can compare this figure with the split tree figure and see that in terms of topology, it all sort of lines up quite neatly. But this is a much more informative figure because you actually have the times and the directionality of all of the recombinations involved. Okay, and what you can actually what you can see is that there seems to be a lot of recombination occurring between this lineage up here that's ancestral to these 0157 serotypes and this clade down here. Now the difference between these ones in red and these ones in blue is that these ones in blue are not STECs, so they're not sugar toxin E. coli, and these ones are. So there's a flow from positive to negative STEC, which is a little bit of a worry, I'm told. And you can detect this more directly, not just from the summary tree, but by looking at the sampled ARGs and computing some summary statistics on the number of nucleotides that are transferred by horizontal gene transfer between these two plates. And you can see that there's an asymmetry between the distribution of nucleotides transferred in each direction, from plus to minus S-tech and from minus to plus S-tech. Okay, so our implementation, as I've said, is called Bacter, and you can, it's a Beast2 package, so you can install it from within Beast2, but all of the documentation and tutorials, etc., can be accessed from this website here. So it performs inference under the ClonalOrigin model. It's precisely the same model that Xavier's ClonalOrigin package does inference under. The slight difference is that we perform joint inference of the ClonalFrame, recombinant edges, and parameters. And in practice, another practical difference is that Bacter can access all of the regular Beast2 substitution models and parametric population models and parameter priors, etc. So we sort of get all of that for free. It also allows you to set up GUI analysis, analyses via a GUI, which is a nice sort of thing that you also get for free in Beast. Now, I don't have a lot of time left, but I'll just say that recombination has, the presence of recombination has some implications for the inference of demographic changes. What I've attempted to draw here is a number of local trees that arise as a result of having conversions in a sampled set of sequences. So here we've only got 2 conversions, but each of these conversions contributes an additional coalescence event. And these coalescence events, these internal nodes here, the timings of them, they tell us information. They're the source of information about the demographic history, so the population size or changes in population size. And the more of them we have, the better we can infer ancestral demographic variation. So this is what was used a number of years ago by Henley and Richard Durbin to infer human demographic history using just pairs of autosomes. So basically sampled from single individuals are enough to infer things like the out of Africa population expansion in humans, which I think is really cool. And we can do similar things in Bacter in a very different way. We can, so Bacter implements a non-parametric demographic history model that's inspired by the standard Bayesian Skyline plot. And in principle, it can reconstruct non-trivial dynamics from very few genomes. In practice, as with a lot of Bacter analyses, it's limited by the computational expense of the algorithm, unfortunately, although we're working on that. So the basic idea is that you split the time between the MRCA of your ARG, or of the ClonalFrame MRCA. You split the time between that and the present up into a number of discrete blocks, and the population size can change at any of those times. So at any of these times here. So there's only a few times that the population size can change. So you wind up with this piecewise constant population model. We also allow you to select a piecewise linear population model as well. Change in population size through time. So here, the dashed line represents an actual step function population change, and the black line represents the mean of the posterior distribution for the inferred population size. And you can see it matches up quite nicely. And you can also see here that although we don't have many samples, we've got a lot of recombination happening. And that it feeds in, if you're able to detect that recombination with good fidelity, then you can use that to improve the inference of this population shift.
Can you say then just a little bit about how the recombination probability ties in with the population size? I mean, I can see the coalescent side of things for sure, but not the other.
Yeah. So I'm not sure exactly what you're asking, but I mean you're asking at which point, because you have a trade-off between increasing amounts of recombination and giving you more information about the population size, and then but if you have too much, eventually you reach this point where you, it's sort of confounding, and you start to lose signal. But
I guess, I mean, it seemed like you were saying that the amount of recombination is informative for the population size calculation, but the direct extension of the Skyline model that I could think of would be more along the lines of just a normal coalescent that wouldn't involve recombination. Or maybe I just misheard you.
Yeah, so maybe I wasn't clear. I meant it's not the rate at which like the number of recombination events that occur in this model that are informing the population size. It's the timing of the coalescence events, be they between whether they're ClonalFrame coalescence events or coalescence events involving recombinant edges, doesn't matter. But the presence of these is the timing of all of these coalescences and the number of them is what we use to the timing of the events is what we use to infer the demographic history. The number of them influences how well we can infer it. So if we have lots of them, we can better infer these population sizes. Is that?
Yeah.
The more recombination, the more coalescence events we have.
Yeah. Thanks.
I mean, and in a perfect world where we could infer everything with perfect fidelity, so we could, if you were presented with the ARG and did an inference of the population size, assuming that ARG was the truth, then it would always be the case that the more recombination you add, the better your inference would get. But unfortunately, we don't observe the ARG directly. We're forced to look at it via sequence data. And it's the more you reach a point, obviously, where you are adding more recombination and making things harder for the algorithm to infer in terms of inferring the ARG. And so that bleeds over into inference of the population size as well. Things are going to get worse eventually. So just to end on a little bit of a downer, there are limitations to the method. Unfortunately, the main one is that the computational complexity scales with the number of proposed conversions. And as I've pointed out, this can be arbitrarily large, even for very small sample sets. I mean, sample sets of size 2. So if you have 2 genomes, you can have arbitrarily many conversions in the history, and each of those result in a fragment of in a local tree that needs to have its likelihood computed. So this can create a lot of it means that your computer will heat up quite a lot while running Bacter analyses. Unfortunately, the usual tricks to speeding up Beast computations don't really apply to Bacter computations because Beast at its core is assuming that the number of trees at least is fixed and the alignments corresponding to each of those trees is fixed. And this assumption is broken by Bacter. So the standard phylogenetic likelihood libraries associated with Beast can't be used, or can be used but don't help, unfortunately. As Xavier pointed out last week, the MCMC algorithm that we use doesn't intelligently locate conversions on the alignment. That's a really good point, and I wanted to be very cheeky and implement a way around this in the last week, but I wasn't able to do it in time. But it's definitely something that I want to address very, very soon. Finally, the network summary algorithm it's a first attempt. As far as I know, there's nothing like this around currently. So we definitely need a lot of research, additional research to find a better summarization algorithm. I mean, finding mean trees or summary trees is already a hard problem and not adequately solved. So it's no surprise that the summary ARG problem is not yet solved, but I'm sure we can do better than what we've done so far. And with that, I'd like to thank all of my collaborators in this research, the members of the Molecular Epidemiological Lab at Massey University in New Zealand, in particular Patrick Biggs and Tessie George and Nigel French for providing the data that we use to prove these methods on. Also the members of the Center for Computational Evolution here at the University of Auckland. They've been very helpful as sounding boards and also in actually collaborating on this research. And I should point out that all of the slides for this talk are available online on my webpage down here. So yeah, if you missed something or you want to look at one of the figures again, it might be easier to go directly to that page rather than the YouTube video. Okay, thank you.
Thank you very much. That was very interesting. I mean, I applaud your energy in taking on like a ridiculously hard problem. Yeah, I mean, just starting from the beginning, I mean, like you mentioned identifiability, and I guess some of these things sort of go away once you take priors on the...
Yeah, that's right. I mean, it's often the case, I guess, in Bayesian analyses that, you know, identifiability isn't the killer that it is if you're doing maximum likelihood analysis. In principle, you can still do the analysis as long as your posterior is proper, right? In which case, you know, as long as you have proper priors, you should be fine. We do, but it still means that the posterior can be extremely broad in different directions. So it becomes less of a sort of philosophical problem and more of a practical implication. It has very strong implications for the implementation of the sampling algorithm. It's still not completely solved either. I mean, we've got these methods that we think are sort of exploring this space, but the distributions as a result of these sort of non-identifiability issues are you know, they're not unimodal distributions at all. They're the posterior surface is extremely complicated, and so with lots of local maxima, etc. So and dealing with that is one of the challenges, and it's part of the reason that the method's so computationally demanding. Not the only reason. There's that, and also the expensive nature of the likelihood computations. But those together, you know, it's you have to be a lot more careful with these analyses, I think, than you have to with regular tree-based phylogenetic analyses, because if you have any prior information that can narrow the search space, you want to use it.
Yeah, like you were talking about, like it would be really nice to have some sort of proposal that that sort of more intelligently chose the recombination tracts. But it seems like that's going to be really hard to calculate. I mean, if you want to do your metropolis Hastings ratio, you have to be able to calculate the probability of these proposals. And if you're using sequence features, that sounds really hard.
Well, no, I don't know. I was imagining doing it in a pretty dumb way, just by looking at the frequency of polymorphic sites along an alignment. And so, for instance, producing the cumulative Hamming distance between pairs of sequences as you step along sites of each of the sequences, right? And you wind up with this monotonic function as you encounter SNPs. And there's going to be sort of inflection points. If you could use the positions of those, if you could hazard a guess at the positions of those inflection points, and then use this to modify your proposal distribution. So with a new conversion, I just basically randomly select a starting point, and then select an endpoint that's drawn from the geometric distribution governed by the track length parameter, right? And so if you just modify that, not in the model at all, but just in the proposal,
okay,
then it could help a lot. I mean, basically, in the extreme case, you'd wind up with this lattice of sites along your alignment that are the only places where conversions can be proposed. And you don't want that extreme case, because there are also that have no signal, and you want to be able to propose them. But to detectable conversions a little bit more frequently, I think, would probably help a bit.
Yeah, I don't know if you feel like bringing the slides back, but I didn't quite understand how the moves work that you described. I mean, is that the one, like you were showing something that was basically showing like a sort of a modification of the clonal frame. Is this the primary clonal frame, like method of changing the clonal frame?
It's one of the main ones. It's one of the main ones that works. I mean, there are actually on the order of 15, I think, different moves that are involved in the sampler, and some of them are, you know, that are ones that actually help during once the algorithms converged, but there are moves that are quite bold that only really get invoked at the start of the chain in the burn-in phase. Yeah, these are the main ones that allow me to basically take Beast's suite of tree moves and apply them to the clonal frame. And this is extremely valuable, because without them, you wind up very easily stuck in these sort of local optima where you know, I mean, you've got the situation that I tried to describe here, where you've got these two different explanations for the data that involve very different topologies, and this is a fairly simple difference between these two. It involves only a couple of recombination events, but you could imagine much more complicated differences that also have almost exactly the same likelihood. That because they involve more than one, it's very hard to set up moves that allow you to step directly between pairs like this. And without such moves, though, you wind up having to get from this one to this one by deleting this edge, then making some changes to the clonal frame, and then proposing new edges. And all of those intermediate states can be very low probability. And so this means that these peaks become quite isolated, and your chain gets stuck in one or the other. So that's what I'm trying very hard to avoid using this sort of approach. It's definitely not perfect, but I guess the way to think about this particular, I know it's, I feel like I'm probably wrong, but it always feels like I can describe this better when I'm using my hands rather than mouse cursor on the screen. But this is basically an SPR or Wilson balding move, right? Where and this is one of the very bold moves that Beast does to explore tree space. Over here, I'm using it to explore ARG space by applying it to the clonal frame. And I'm taking the edge above, so I'm randomly selecting some edge in the clonal frame, in this case C, and I'm detaching this edge C and reattaching it down here at this point just above B. And in so doing, I have to deal with the fact that I've got these 3 points here, or 4 rather, that these 4 nodes that are on this ancestral edge that I'm about to delete. And my approach is to take those attachment points and ensure that they still remain ancestral to C. So attach them, in this case, on the line that joins on the lineage that is going to be ancestral to C after I've made the modification. So which includes all of this black line over here. So I'm shifting this green this green node over here, this one over here, and I would shift this red one over here as well, but this is going to violate the model because it actually marginalizes over events like this, so they can't be included explicitly. Ones that loops that appear above the root of the clonal frame. So that one needs to be deleted. So that's the process of getting from here to here. And it all just happens in reverse when you go in the other direction. But it's not. There are, you have to flip a coin basically to decide whether to, when you detach the C and reattach it over here, you have to decide whether or not to move the points, these connection points that were ancestral to C over to the new point, because obviously not all of them can move, otherwise it's not going to be reversible. I'm not sure if that
yeah, no. I mean, yeah. I mean, you almost wonder though if like maybe it would be possible to, like, somehow these like recombination nodes, maybe they could be suggesting locations for your clonal frame to change, because if you have one of them that's sort of suggesting there's some additional relatedness between 2 different branches of the tree, so you could like swap out like turn the ClonalFrame edge into a recombination edge, and then
Yes.
a recombination edge into a ClonalFrame edge.
Absolutely. That is another strategy, and I have implemented something like this.
But it doesn't work.
No. It does. It's tricky. One of the things about MCMC that make it an art, right, is that you can, in principle, all you need is for your operators to your proposal distributions to produce a walk that is capable of exploring the entire state space, and anything that you do beyond that is never really going to hurt. So you can have moves that don't actually do a lot for most of the time, but when they are needed, they are extremely important. So this ClonalFrame recombination swap operator is one of those that isn't accepted a lot, but it does seem to improve the ability of the sampler to get between these peaks. But yes, that's it's a very tricky thing to think about, though, because it's not how should I put it? It's not always possible to pick some recombinant edge in this model and say, all right, I'm going to swap the role of the ClonalFrame and the recombination for that edge.
Because there's all the other edges, and you have to take care of.
Because there are all the other edges, and it interacts with everything else. So you can sort of do it, but not do it exactly.
Awesome.
Yeah, that's a good idea.
Well, we should wrap up. We're sort of well over time, but that was very interesting. Thank you, Tim. So that wraps up our bacterial phylogenetics session. And next, we're going to have a philosophy talk about Gaia theory from Ford Doolittle. So he is well known in the bacterial world, but he's going to be giving a philosophy talk. So that will be interesting. All right. Thanks, Tim.
Thanks, Eric.